Okay, next, right, we will be looking at some application of what you have learned so far in epistasis. We will look at how epistasis can be applied in enzymatic pathways, okay? So if you recall, right, enzymatic pathways are pathways where you have more than one enzymes which work together to form a particular product or work together in a particular reaction. Okay, so my example in the notes is a reaction to produce different colored pigments, right? So we have the yellow pigment, you have the red pigment. Okay, so in this enzymatic pathway, what you see is there are two enzymes involved. There's also an inhibitor, right? So all these enzymes and inhibitor, right, are coded for by genes, right? So since the enzymes work together in the same reaction, they interact together to form the colored pigments, right? Then likewise, right, we have the genes that codes for these enzymes interacting together, right, to give you different color, right? So genes interacting together are gene interaction and epistasis. All right, so in this case we have the second step, right, in the pathway, which is dependent on the first step of the pathway, right? So if the first step does not occur, right, then the second step wouldn't occur as well, regardless of whether we have active or functional enzyme Q or not, right? So can you imagine that enzyme P is somehow non-functional, where maybe the gene codes for a non-functional enzyme P, or we have the presence of inhibitor B, right? Then enzyme Q, whether it is functional or not, the color of the organism, right, will still be white. So you see some masking here, right, in the enzymatic pathway, right? So when the enzyme P is non-functional, it masks the expression of enzyme Q, right? So whether we have enzyme Q or not, we still wouldn't get red pigment, we won't even get yellow pigment, right? As long as enzyme P is not active, right, it will be white. Okay? Right, so we will see some examples later to make it clearer, okay, to help you understand it better. Okay? So in this example, right, we are looking at the three genes involved, right? Two genes coding for the enzymes and one gene coding for the synthesis of the inhibitor. Okay, but at any one time we just look at two genes, because to look at three genes together would be too complex, right? We don't want to try hybrid inheritance, right? We just look at dihybrid. Okay, so firstly, let's assume that our inhibitor B is not synthesized, right? That the genotype of the parents is small p, so we don't consider the inhibitor. Assuming the inhibitor is absent, we just look at the enzyme P and enzyme Q, right? So we have a double heterozygote parent crossing with another double heterozygote parent. So what would be the phenotypic ratio, right, for this cross? It would be 9:3:4 if you work it out, because the small p genotype would be epistatic over gene Q. Right, small p, meaning that there is inactive enzyme P, right? So like I said earlier, when enzyme P is inactive, it masks the expression of Q, right? Because you will be white as long as enzyme P is inactive, regardless of whether we have active or inactive enzyme Q. Okay, so to put it in a genetic diagram, of course you should draw it yourself. Okay, and you can check against mine if you want to, but anyway, this is the diagram that I've drawn, right? You can see that it is a 9:3:4 inheritance, or rather 9:3:4 phenotypic ratio, because of the recessive epistasis by small p genotype on gene Q. Okay, so 9:3:4. So in the same example, now we assume that enzyme P is active, right? That or the active enzyme P is produced, okay, and that the genotype is just homozygous dominance for P, right? So do we ignore gene P? We just look at the inheritance for gene B for the inhibitor and gene Q, right, for enzyme Q. Okay, so we have double heterozygous, again, crossed with another double heterozygote. What would be the phenotypic ratio? If you draw the diagram, you might want to pause now and draw the diagram and try yourself, all right? But anyway, I will just go on, right? So the ratio would be 12:3:1, which is the ratio for dominant epistasis, right? Because for the gene B, right, when it has the dominant allele, whether homozygous dominant or heterozygote, with the dominant big B, right? Inhibitor B is synthesized, which inactivate or inhibit enzyme P, right? So with the synthesis of inhibitor B, it would be white, regardless of whether active or inactive enzyme Q is synthesized. Right? So if you have drawn the diagram, you can check against mine, right? It is dominant epistasis due to the presence of the dominant big B allele, right? So 12:3:1. Okay, so we have just looked at the two epistasis that we have covered in the previous video, right? Recessive and dominant epistasis applied or seen, observed in a particular enzymatic pathway, right? So that's the that's the theme of this particular video, right? Application of epistasis in enzymatic pathway. All right. So we're going to look at another type of epistasis that we can see in enzymatic pathway. This is known as complementary gene action. Right? So if you look at the example that we have here, there are only two phenotypes, right? Either white or purple, right? For the color, okay? So either white or purple. You need both enzymes R and G in order for the phenotype to be purple. When there is no enzyme R, it will be white. Compound A will stay as compound A, which is white. Or if we have enzyme R but no enzyme G, then it will be also white, because compound B is also white. You need both enzymes active, right, for compound C purple color to be produced. Right, so two genes, the two active enzymes give you one phenotype. Absence of any one or both active enzymes would be white. So this is known as complementary gene action. Okay, so following the same example, if we do a cross between two double heterozygote, right, you can again draw the diagram yourself and try. Right, it's basically the same as a normal dihybrid cross, but then the only difference is when we have the phenotype for the offspring, right? As we have seen for the other epistasis. Okay, so you will get a 9:7 ratio. Okay, so if you have drawn the diagram, you can look at mine and compare, right? So double heterozygous cross with double heterozygous, you should have the 9:3:3:1 if it is Mendelian, but in this case, because of epistasis, these three together, the 3:3:1 together 7, would be white, right? This is white, absence of both enzymes or both active enzymes. In this case, we have absence of active enzyme R. Here we have absence of active enzyme G, right? So absence of either active enzyme or both give you the same white color. Right, you need both active enzymes for purple in our example. Okay, so this is a complementary gene action that you can also see in a enzymatic pathway on top of the dominant and recessive